Thank you very much. So this is not advancing yet. Oh, now it is. Okay. So as I mentioned, I work at the interface of chemistry and biology, and one of our key questions is how cell heterogeneity gives rise to functional impact in terms of biology, right? What is the importance of cell heterogeneity in terms of biology during states of health, during states of disease? Now, we talk a lot about measuring a large numbers of cells because obviously we want to live up to perhaps to transcriptomics, but we should not forget about one important thing is that cell heterogeneity is important as long as you know where and when cell heterogeneity is defined in a particular biological context. And that is very true during the development of neural tissues, the nervous system. So take, for example, the early lifetime. All of us went through these processes, the developing vertebrate embryo, as you can see here, dividing into 4, into 8, into 16 cells, 32 cells, 64 cells, and division goes on and on. Every single cell is different, actually. In this particular model called the South African clawed frog, which is what my lab uses. Yes, it's an actual frog that has claws. We can track the tissue fate of every single cell from the 16 and the 32 cell embryo. So there's a huge power in terms of understanding how not just cell heterogeneity evolves, but how cell heterogeneity gives rise to these functional differences at the level of tissue development. And then at the other side of the spectrum, in terms of the nervous system, we also want to understand how individual neurons that are located in different areas of the cortex, of the hippocampus, various areas of the brain, differ from each other. And not just because of proteomic differences or metabolomic differences, but also because of the functional differences that they do in terms of neuron-to-neuron communication, processing, information from outside of the biological system, and so on. I don't need to convince this audience about the importance of single-cell measurements, and Nikolai very nicely alluded to the importance earlier in the morning session. But I just want to mention one more thing again, that besides measuring individual cells, we also ideally need to know where exactly and when exactly in a particular model those single cells are collected and analyzed. And so although it's possible to dissociate tissues into large numbers of cell suspensions, by dissociation, we by definition lose that information. And so what today I'd like to tell you about is how my laboratory is trying to overcome some of these challenges while making an important statement. We're not taking advantage of any of the beautiful advances that Ryan, Nikolai, and the other laboratories have introduced in terms of handling very small amounts of materials. We're still using old school Eppendorf vials, and we're still processing these single cells accordingly. So we have a lot of protein losses we're encountering, and we hope to very soon adapt some of those technologies. So to understand cell-to-cell differences, we now can characterize genes and transcripts, but this is really where the functional differences are now in terms of focus. At least particularly in my lab, proteomics, spectromics, and metabolomics. My lab has tackled these 3 areas. Today, I will really just focus on proteomics. So what do we do in the laboratory? To understand cell-to-cell differences, we work with the frog embryo, we work with the mouse, central nervous system, and even zebrafish. These are our biological models because they are used in different biological contexts. Then we build mass spectrometry instrumentation to enable the ultra-sensitive detection of proteins, peptides, and metabolites so that we can identify, characterize the molecular phenotype of the particular cell of interest. And then we don't just stop there, because then we want to loop in the biological insights. And how do you do that? Well, now you need to go to biology and literally recruit what biologists have been doing for decades and centuries. And so we basically use high-resolution microscopy systems capable of fluorescence imaging in particular cases, various translation stages, microinjectors to be able to access single cells, to inject particular molecules molecules into them or to withdraw molecules from identified cells. We also do electrophysiology, particularly on single neurons in communication in the mouse brain. And we even do behavioral assays, how an entire organism, for example, the swimming tadpole, will respond to perturbations at particular single cells during early stages of development, because I believe it's the integration of chemistry and biology is what's going to drive us into the future. So in these models that we actually pick strategically, the cells differ widely in size from 250 microns giant cells down to 10 microns somatic cells. Now, this large cell is not less important because it's big, right? This is actually how we all started as embryos. And if we didn't have the right size to then divide, we would not be here. So it's not just the size that matters in terms of biology, right? It's actually the biological context. Now, these cells obviously will contain very different amounts of materials. And so it's the integration of biological chemistry and then applied biology that you will hear about a little bit today. Now, I will shift gears from liquid chromatography. We do liquid chromatography. It's a great technology, but there's another technology in the field, and we did not invent this. This is capillary electrophoresis. It's an emergent technology, and I really advise everyone to keep that in mind as you're planning your next generation single cell measurements. So my laboratory built this interface where we're able to deposit about 200 nanoliters of liquid, sample, protein digest, metabolite extract, whatnot, using old school pipettes and a good magnifying glass, right? And now we are able to position this thin capillary, 110 microns OD fused silica capillary, over into the sample. Then we have way to actually inject 1 to 10 nanoliters of that sample into the fused silica capillary. And then we remotely operated a high voltage power supply to apply up to 30,000 volts across this fused silica capillary filled with an appropriate electrolyte so that we can separate by electrophoresis the charged biomolecules. What are the charged biomolecules? If you want to study proteins, then proteins. If you want to study peptides, then they're peptides. If you want to study metabolites, then those are metabolites. They separate as long as they are charged. Kind of like liquid phase mass spectrometry, jokingly, I usually say. But anyways, now the biomolecules migrate, and we also custom built capillary electrophoresis electrospray ionization interfaces and very unique operational modalities so that we can efficiently ionize those biomolecules. We have heard a lot about the importance of using electrospray ionization to the best of our capabilities. This is one way of doing it by maintaining the stable cone jet spraying regime. And then we use different kinds of mass spectrometers depending on the type of questions we've got. So over the years, we have optimized microsampling, separation, ionization, detection, analysis in terms of data analysis to get to the deepest coverage that we could while still using old school Eppendorf vials for sample processing. These platforms that we actually build are quantitative, both for small molecules and proteins and peptides, because we can map out concentration calibration curves using authentic chemical standards. So for metabolites, we were able to go down about good number of years ago to 25 attomoles with about 10% of quantitative error. And for peptides, we were a couple years ago at about 210 zeptomoles of sensitivity. That's 126,000 copies of a peptide. That's kind of what you need when you're going to go after single cells, right? You need high sensitivity. And so my laboratory over the years have learned these tools. We have kept building more and more of these platforms. This was our first generational platform with a C system shown here, coupled to a quadruple time-of-flight instrument. Then we built another CE system coupled to a quadruple orbit trap mass spectrometer. It's now at 200 zeptomole sensitivity. Then we built another C platform to couple it to a tribrid Lumos platform for even higher sensitivity. And more recently, we built a 4th generational platform where we use CE on a trapped high mobility time of flight probe mass spectrometer. And most recently, and this was just released in the literature, so you will be able to find, we actually came up with different ways to even boost the sensitivity further. It's really a name of the game in our field is sensitivity to some extent. So here what we are able to do is we are able to load much more material into the capillary than historically capillary electrophoresis would allow. We normally would just load 1 to 10 nanoliters of sample. That's wasteful if you actually have maybe a microliter of sample available to you. So here now we could load 10 times more sample via a process called large volume sample stacking, where we use something called a dynamic pH junction, where we play with the isoelectric point, with pH conditions, so we can enrich our compounds directly in the capillary, so that we can boost the abundance of the peaks for better sensitivity, identification, quantitation, and so on. And then we use a timsTOF Pro instrument for detection. Now, we actually use this platform on the paravalbumin nucleus of the mouse, which is a very important regulatory nucleus in the central nervous system, controlling thirst if you're thirsty. That's why, or if you're stressed out, that's actually one of the reasons. And what we found that when we developed this platform and then used timsTOF Pro, we gained a lot of improvement in terms of sensitivity. So here are representative numbers in terms of signal-to-noise ratios. So compared to no ion mobility, black, the gray curves are with ion mobility, trapped ion mobility. We got about 5 to 50-fold improvement, in part because of ion trapping, probably, in terms of sensitivity. And that allowed us to still demonstrate quantitative performance using this large volume sample stacking approach. But now we were able to go down to 30 zeptomoles of sensitivity. That corresponds to 16,000 copies of a peptide, or a protein, if you will. All right, so that's great. And now we've got this tool that now we could apply in different contexts. So I will show you a couple of examples without really going into the biology. If you're interested, I'm happy to talk about them during the break. So this is one example where we can take an embryo that's developing every 15 minutes, literally in front of our eyes, where we're able to identify the cell, exactly where in the embryo is, what particular developmental time point in the embryo, so that now we can go into that single cell using fabricated microcapillaries that we translate using a 3-axis translation stage, and then we aspirate into that capillary from the cell content using another instrument called a microinjector connected online. So now then we do the sample processing, again, old school Eppendorf vials at this point. I wish we could use nanopots and the other advances. So what's good here is that you can do these measurements for peptides, to do proteomics, biomarker proteomics, but also for metabolites. And that means now we're at the point where we are merging together both proteomes and metabolomic information to get to a systems biology understanding of single cells. We're able to actually represent various pathways of canonical knowledge. We're able to reconstruct actual pathways where both proteins and the metabolites are detectable, are quantifiable. That's what you want to have if you want to really study biology, right? You need to know where a particular pathway is heading during states of health or disease. And so we've actually used this information to discover cell-to-cell differences of developmental significance in the embryo, all of which I will skip over at this point. More recently, we developed an approach in which we were able to aspirate such small amounts of material from these identified single cells in live vertebrate embryos from the frog that the actual embryo just healed its single cell membrane and then continued normal development. The cell just continued developing like nothing happened as far as we could tell, to the point that we actually were able to culture embryos up to larvae and even behaving tadpoles. right? So that means we could now, with one sampling, analyze the content of the proteome in that single identified cell. This is a cell that reproducibly actually gives rise to neural tissues in the model. And we could also then use the metabolomic platform that I've showed you earlier to characterize this dual proteome metabolomic profile of that single cell. And then once we got to the larval and the behaving tadpole stage, that's where the biological characterizations come in. We can characterize anatomy, we can characterize behavior. For example, we have used these tadpoles in various background color preference assays to measure their visual and muscular performance. And we actually find that this approach does not perturb any of these characteristics that we could find. So this is essentially one of the first examples I'm aware of where we not only can do single cell dual proteome metabolomics at live embryos, but the embryo survives the analysis. So this is based on in vivo mass spectrometry approach, with the caveat that we're still aspirating out something from that single cell, right? It's not like optical microscopy. But then now you've got the biological phenotype, and now you've got finally the dual proteome metabolomic phenotype of that single cell. So these are large cells, and you may not be interested in embryonic cells, and that's fine. You might be interested, though, in actual neurons or smaller cells. So we wanted to know whether this whole capillary microsampling approach is downscalable. So we started measuring different amounts of proteins from cell cultures, and basically we're able to identify a good number of proteins that were very encouraging to then allow us to move forward into cultures, actual neural cultures, where we could use this capillary to literally pick up a single cell located there, for example, from there. And then now you see that cell right in there. I don't know how much you can see from the distance, or virtually that cell actually is getting lysed literally in the capillary. We were able to identify a couple of like 50, 100 proteins actually from that single cell. So that's cool, but that's still neural culture. Can we do this in vivo? And so we actually worked with a neuroscientist to section the substantia nigra of the mouse central nervous system. We then moved to an electrophysiology setup where we could record the electrical activity of live single identified neurons in the mouse brain. We could tell, for example, that that is a dopaminergic neuron, that is a GABAergic neuron, and then we used that patch pipette to then aspirate the content of the neuron after electrophysiological characterization into the pipette, followed by with our protocol to then characterize the proteome from that soma of the neuron, right? We're actually only collecting a very small portion of the neuron. We're only collecting or analyzing 0.25% of the total protein content of the soma. Soma is a cell body of the neuron before the dendrites and the axons, all right? So we're measuring a picogram of protein. It's very small, right, compared to what historically we have done. So just to give you an idea what you can do now with these tools. So in this example, we took 3 different dopaminergic neurons that we identified and characterized electrophysiologically in the live mouse brain. And then we piggybacked on various strategies, including the quantitative carrier approach from Nikolai's laboratory, carrier proteins from elsewhere to identify about and quantify about 150 proteins from these single neurons. And the neuroscientists are telling me that these results make sense because we're actually identifying proteins that are known to be expressed in dopaminergic neurons. Some of the genes are located here. Now you can do all kinds of gene ontology and pathway analyses to link this into actual functional biological studies. I'm skipping the biological applications, but we have publication along those lines. Then the 4th generation approach I mentioned you earlier now actually allowed us to boost these identifications to about 700 different proteins from single neurons while only measuring about 0.25% of the proteome content of that single neuron, thanks to capillary electrophoresis, mass spectrometry. And these are some of the proteins that apparently are important for neuroscience. The lower abundance domain actually includes transcription and translational regulators. So that's cool for biology if you're after health and disease studies. And I just want to show you how you can even aspirate the entire content of the soma. So this is a transgenic mouse line we use, where we're labeling in green fluorescent protein the dopaminergic cells from the substantia nigra of the mouse. So you see these lighting up patches. Those are the actual neurons. And then we go in with our capillary. We aspirate out the entire content of the neuron. That whitish streak is the fluorescent signal that's being aspirated, sucked into the capillary. And I can show you this, the aspiration was successful because, well, the GFP signal is gone at this particular location, while all the other neurons are still there. Right? So you've got the landmarks to line them up. So successful collection of the entire neural proteome, as we could tell based on the GFP signal, is accomplishable using this patch pipette approach that we developed a couple of years ago and recently also published, while concurrently being able to record the, for example, a slow pacemaker pattern of that dopaminergic neuron. Now you can think about degenerative diseases, right, to go after. You can do pharmacological manipulations to these particular neurons if you've got a particular study, while not only measuring the physiological behavior, but the proteome content. And most recently, just we, for fun, thought, could we actually use this approach to ask whether we are able to find proteomic differences between different neuron types that are spatially and temporally identified using also electrophysiological information in the mouse brain. And so we picked the dopaminergic neuron class, a paravalbumin interneuron type, and a serotonergic neuron type. We were able to analyze about a picogram of each of these proteins. We still have a lot more protein actually we can measure. We're kind of wasteful, to be upfront. We're identifying anywhere between 200 to 800 different proteins. And now comes the cool part. Once you ask whether quantitatively the protein abundances are different between these cell types, that's actually what we found, that yes, that if you actually do a hierarchical cluster analysis on normalized data, then all the sample types that are dopaminergic cluster together, but cluster differently from the paravalbumin neurons, that also cluster differently from the serotonergic neurons, solely based on the proteomic content. So it's kind of cool to think about this, that the soma, the cell body of that neuron, is already indicative of the phenotype of the neuron. Right? And this is just the beginning of where now you can push this technology, because if you now have a proteome information on the neuronal content, you now can open the door to a proteomic-driven classification of cell types in the brain, not just mouse, but maybe other vertebrate organisms too. So in conclusion, so single cell development is really a possibility at this point, and many of us are now beginning to use these technologies. And here, really, it's a big hands up to all the laboratories that are coming together and sharing their knowledge to make these measurements possible. Neither of us would be able to do this on our own. It takes technology, it takes bioinformatics, it takes specialized data acquisition strategies to move us forward as a community. My lab has focused a lot on instrument development, and you can see some of these instrumentation. We're now beginning to actually build automated systems, and I hope to report on that soon. We've used these tools to ask very specific biological questions, none of which I really talked about. We have discovered molecules, for example, that can alter cell patterning in the vertebrate embryo, cell-to-cell communication in the embryo, just because we've got these novel data that are not historically available in the literature. And this really opens exciting new opportunities, not just in analytical chemistry, but really biology and translational research forward. So we have a really cool community coming together to that end. So I want to thank my research group. These are old pictures pre-COVID, who really are the heroes here, who did the hard work, and the various funding agencies who actually believe that these kinds of developments and applications are possible. It's not easy to break through into new directions, especially not without sufficient funding. So I thank the funding agencies and the peer reviewers, of course, who believe that these proposals are feasible. Last and not least, thank you for your attention. If you have any questions, I can take them now or during the break. Thank you.
Thank you so much for that talk. We really appreciate it.
No problem. If there's time, I don't know if I went over.
We have some time. You went over some of the time for questions, but we have
okay.
more. We like questions. So this is Nikolai Slavov asking a question. So my question, Peter, is what are your thoughts on coupling capillary electrophoresis to an autosampler?
Yeah, that's precisely where we would like to head, right? Because right now we're doing sample loading completely manually. So basically what that means is somebody actually is literally squatting in front of this platform and operating it. It's 1 cell per hour throughput, right? I don't wish it on my enemy. Fortunately, my lab is agreeable to do these measurements. But yeah, automation is definitely the way to go. Similarly to how nanopods-based automation is actually moving us forward.
So this is something that you would develop in your lab as specialized equipment, or you would also team up with companies to make it accessible to others?
Right now we're doing it in our lab, but if there are companies who would like to join us, we would be very glad to have your support. Yes.
Thank you.
Point.
Just a couple of quick questions. What flow rate do you usually use? Yeah, so CE is weird if you come from an LC background because there's no actual net bulk flow through the capillary if you do CE correctly. The actual compounds migrate at about a nanoliter per minute of flow rate. Ideal for nanoflow ESI, if you actually think about it. So C is one of those, I think, rare technologies that just naturally integrates well with ESI, as long as you know how to make that work. And that's really some of the challenges we face in the field. Alexander Ivanov has done a really nice study actually demonstrating that if you do these couplings right, you can get to really deep proteomes in relatively short analytical timeframes using capillary electrophoresis mass spectrometry. So we were not pushing the liquid through the C capillary. And then those pipettes, the tips to get into like a neuron.
Yeah.
They're really tiny.
Yeah. So we pull them to about, well, it depends on the cell, right? If we have an embryonic cell that is 250 microns, that's going to be more forgivable to a larger ID capillary. We usually go with 50 micron OD capillaries for that. For single neurons, I think we went down to like 10 microns or 5 micron IDs, ODs, something like that. Yeah, it's all scalable, right? That's kind of the title of the talk. Depending on what cell you're after, you pull the capillary to the dimension that you want to accomplish. Yeah. Thanks, Ryan.
This is Bori Petrova from Children's Hospital. I want to get your ideas on how you think about heterogeneity, but from the perspective of same cell type. Especially you've done proteomics and metabolomics. Is there a difference between the two, and in what way? And what is the throughput you would need to capture that heterogeneity? Yeah,
very good question. So we early on asked whether the proteomic differences we actually detect between identified single cells in the embryo are detectable at the level of transcriptomes, and the answer was not. That was surprising. So the proteome actually revealed cell heterogeneity in this particular model, for which a lot of these sequencing experiments have been done, that transcriptomics data did not uncover. Same thing applies for the metabolome, actually, that we are able to find cell-to-cell differences, sometimes better at the level of the metabolome than the proteome. But it's not like any of the omic levels is better than the other. I do believe an integrative approach is really what we want eventually, right? The more orthogonal types of information at the level of molecular architecture we have about the cell, the better we understand how those cells differ from each other. Yeah. Some neurotransmitters, for example, are different. Some small metabolites are different between the cells that the proteome did not even indicate to us.
Good question. Thank you. Hi, Andrew Leduc from Northeastern. Great talk.
Thank you.
I had some questions about what type of metabolites are, I guess, for doing liquid chromatography, you need to, use different gradients and such to capture different sets of metabolites. So is this something that capillary electrophoresis, you can kind of, are there different techniques, I suppose, to get different metabolites, or can you kind of capture a large array of them? And are there certain ones that you're used to looking at? Since the space is kind of large.
Yeah, great question. Thank you for asking it. So because we do CE, we don't worry about packing material stationary. There's no stationary phase in our systems, right? We're not doing retention chromatography. We're doing electrophoresis-based separation in these cases. And so as long as the compound, the metabolite is charged in the liquid phase, it will migrate under the influence of electric field. Right? Now, the question is, if you are a cation, if I may ask you to be a cation, right? And you apply large positive potential on the inlet end, then as a cation, you're going to move away from it to the outlet, right? But if you're now an anion at the same condition, you're going to move backward. So that's kind of cool because that means you can now quickly switch polarities to move anions or cations during separation, or you can play with the pH of the electrolyte that is in that capillary, and then alter the charge state of metabolites, peptides, proteins. Same thing applies, right? We've done both anionic, cationic metabolomics, and we get nice orthogonal coverage of the detectable portion of the metabolome. The separate conversation, identifying metabolites is a whole different conversation than identifying proteins, but we can talk about that, yeah.
Amazing tool. Thank you very much. I was wondering, do you use sheet liquid or sheetless system?
Oh, very good. So you work in CE. So we actually use sheath-based, and this is a relatively large sheath flow that we used to do back in the day. Now, like a microliter per minute sheath flow, which is pretty big dilution, right? Now we went down to about 100 nanoliters per minute, which is still a dilution in the Taylor cone, but because we are operating the spray in this very particular regime, we kind of make up for the lost sensitivity, I believe at least. The lower we can go with the sheath flow, the better we will be off. And there are sheethless interfaces, right? So there's a lot of room actually for CEESI to give us even better sensitivity than my lab has demonstrated over the last about a decade. Yeah.
And do you apply top-down experiment?
Top-down?
Yeah.
So I haven't done that yet in the lab. We are actually getting into that. Liang's son has done very nice CEESI-based top-down measurements. Yeah.
Thank you.
Yeah.
Alexander Ivanov, Northeastern University. Great talk, Peter. Thank you as always. Since you mentioned that first, I was not going to ask about that, but capillary electrochromatography. What do you think about that and the future potential?
Yeah, it's a lot.
A discussion at the SMS, so I just would like to follow up.
It's actually a lot on my mind too. So there's a very particular way of operating CE where you're creating conditions so that you're moving the bulk flow through the capillary without creating parabolic flow profile. Capillary electrokinetic chromatography is that technology where now you can add in a stationary phase also, like a packed column, for example. So that would be a merger of LC meets CE. We, in fact, today had conversations about challenges with pumps maintaining a very low flow rates. This is a way around it. And all you need is a high voltage power supply, right? Just energize it, right? And then you have appropriate, fused silicon capillary chemistry to maintain the electroosmotic flow. You use that to move the liquid through. So that's a very exciting direction. Are you working on it?
Oh, have you?
Okay.
I have worked on that some years ago, but what about you?
Yeah, it's a very, very intriguing direction. There's even micellar electrokinetic chromatography where you can dynamically create stationary phases.
Hi, this is Hannah Wilkins from Johns Hopkins. Thank you for the talk. It's wonderful. I just had a question with the capillary. Have you quantified a baseline or have any idea of like if there's any sample that is sticking to the capillary or anything like that?
Oh, very, very good question. Yeah, so indeed that can happen. If you've got cell lysates, they're dirty, and that just came up in, where are you, Ryan's talk, right? So that definitely can happen. Some proteins actually can adhere to the capillary surface if you're not using the proper chemistry coded capillaries, which we are not in these experiments. We have ways to actually check for that. So we can monitor migration times. We don't call it retention time, we call it migration time in capillary and if our migration times start deviating, that's usually what happens. The capillary gets coated by sticky stuff, for lack of a better word, but we can quickly recover that by sodium hydroxide based conditioning of the capillary. We just etch off the stuff, and then we're back to the game. That's how we've been managing. If you're doing top-down proteomics, though, you're going to want to coat the capillary with certain chemistries so that your proteins are not going to be lost on the surface.
Cool. Is there any other questions before we move on to the break?